it's a little bit more complicated than that because there's more than one primer that they can use. So this whole testing thing is very, very heterogeneous around the world. So in other words, there are different protocols in virtually every country. And a lot of them are recommended through the World Health Organization, but there are some from other bodies too. And they can use different primers. So, and even when they do just one sample, they're not going to use just one primer for a test and say if that one's positive, it's positive. So it's not exactly that clear cut. But what we're talking about here is at least one of the primers that they're using in one of the tests for our own sequence. Okay, so this could come up positive in any human, regardless of the presence of any virus or anything else. So it adds another level of confusion and obfuscation to how do you interpret the results of a test like this. Now, I still think the most salient point is that we don't know where these sequences came from because there was never a purified particle of a virus where they had a bunch of them together in one sample and they could characterize those and pull the RNA material right out of those particles. They never did it that way. They only looked at it in this dirty sample, which was a mixture of many, many microorganisms and human cells. And they pulled these sequences out of there because they thought the sequences belonged to the family of coronaviruses. But that was based on only about 80% sequence identity between those samples. And, you know, when I looked and did that research previously with the other probes for a different protocol, for the Germany protocol, I found that many of those primers in that sequence had 80 to 85% sequence identity with stretches in the human genome as well. I just didn't find a complete match. I did find 18 in a row that matched, but those primers were 24 bases long. So there's a lot of overlap in this. And then if you also look at the nature of the PCR test itself, is that the enzymes that they use to copy the RNA make mistakes. And so they could give you a lot of false positives. And this is one of the problems that's always been known about this test. And the more you amplify it, because you do this test in cycles, right? And each cycle you double the copies of that piece of RNA that you're looking for. And the more times you double it, the more false signals that you get, because you're also amplifying like the noise signal, which could be other sequences that are partially matched, and they're not detectable until you amplify them so many times. And then it's like everything that is an artifact in there is also detectable, and that gives you a false positive. And that's one of the main reasons why Carrie Mullis did not recommend using this as a diagnostic test, because of all those false positives. But, you know, in my opinion, since we don't know what the origin of this sequences that they're testing is, other than now we know one of them may come from our own sequences, like the human sequence, then there's really no way to calculate any accuracy. So I would say, you know, there's 100% error rate with this test.